Okay, myself, Dr. Prashant Upadhyay, I am Associate Professor, Department of Pharmaceutics, School of Pharmaceutical Science at IFTM University. Today I will teach you about something about Biopharmaceutical Classification System. In the introduction part, BCS has been developed to provide a scientific approach to allow for the prediction of in vivo pharmacokinetics of oral immediate release drug product by classifying drug compounds based on their solubility related to dose and intestinal permeability in combination with the dissolution property of the dosage form. The importance of drug dissolution in the gastrointestinal tract and the permeability across the gut wall barrier in the oral absorption process has been well known since 1960, but the researchers carried out to constitute the BCS has provided new quantitative data of great importance for modern drug development, especially within the area of drug permeability. According to BCS, drug substances are classified as follows: Class 1 high permeability, high solubility; Class 2 high permeability, low solubility; and Class 3 low permeability and high solubility; and one more Class 4 low permeability and low solubility. The BCS is a scientific framework for classifying drug substances based on their aqueous solubility and intestinal permeability. When combined with the dissolution of the drug product, the BCS takes into account the three major factors that governs the rate and extent of drug absorption from immediate release solid to oral dosage form, that is dissolution, solubility, and the intestinal permeability. Based on class boundaries, there are various classes. A drug substance considered highly soluble when the highest dose strength is soluble in 250 ml of water under the pH range of 1 to 7.5. A drug substance is considered highly permeable when the content of absorption in human is determined to be above 90% of an administered dose based on mass balance or in comparison to an intravenous reference dose. A drug product is considered to be rapidly dissolving when more than 85% of the labeled amount of drug substance dissolved within 30 minutes using USP apparatus 1 or 2 in a volume of less than 900 ml of a solution. Now consider the benefits of knowing the BCS category of a compound. What are the benefits? It can save both time and money. If the immediate release orally administered drug meets specific criteria, the FDA will grant a waiver for expense and time-consuming bioequivalence studies. The aim of the BCS is to provide a regulatory tool for the replacement of certain bioequivalence studies by conducting accurate in vitro dissolution tests. Other benefits: the steps will certainly reduce the timelines in the drug development process, both directly and indirectly, and reduce unnecessary drug exposure in healthy volunteers, which is normally the study population in BE studies. It has also been reported that the application of BCS strategy in drug development will lead to significant direct and indirect saving for the pharmaceutical companies. The BCS has been developed primarily for regulatory applications, but it has also several other applications in both the preclinical and clinical drug development process and has gained wide recognition within the research-based industry. These are some other benefits. Combined with the dissolution, the BCS takes into account the three major factors governing viability, biodissolution, solubility, and permeability. This classification is associated with the drug dissolution and absorption model, which identifies the key parameter controlling drug absorption as a set of dimensionless numbers. Some key parameters controlling the drug absorption. First, the absorption number. It is defined as the ratio of mean residence time to mean absorption time. Second is the dissolution number, defined as the ratio of mean residence time to the mean dissolution time. Third, the dose number, defined as the mass, that is dose, divided by the product of uptaken volume, and the solubility of the drug. Class 1 drug exhibit a high absorption number and a high dissolution number. The rate limiting step is the drug dissolution. If dissolution is very rapid, then gastric emptying rate becomes a rate determining step. For example, in the case of metoprolol, deltiazepam, verapamil, propanol, hydrochloride. Class 2 drugs have a high absorption number but a low dissolution number. In vivo drug dissolution is then a rate limiting step for absorption except in a very high dose number. The absorption for class 2 drug is usually slower than class 1 and occurs over a long period of time. In vitro in vivo correlation, IV/IVC is usually accepted for class 1 and class 2 drugs. That example phenytoin and ketoconazole, etc. For class 3 drug, permeability is the rate limiting step for drug absorption. These drugs exhibit a high variation in the rate and extent of drug absorption. Since the dissolution is rapid, the variation is attributable to alteration of physiology and membrane permeability rather than the dose form factors. Example, cimetidine, acecloprid, neomycin, captopril, etc. Class 4 drugs exhibit a lot of problem for effective oral administration. Fortunately, extreme examples of class 4 drug compounds are the exception rather than the rule and are rarely developed and reach the market. Nevertheless, the number of class 4 drugs do exist. Examples such as Jaxol. This is in vitro in vivo correlation shows how the in vitro release studies and in vivo studies are being correlated graphically. After this, what is the application of BCS in oral drug delivery technology? Once the solubility and permeability characteristics of drug are known, it becomes an easy task for the research scientist to decide upon which drug delivery technology to follow or develop. For class 1 drug, the major challenge in the development of drug delivery system is for class 1 drug is to achieve a target release profile associated with the particular pharmacokinetic and pharmacodynamic profile. Formulation approach include both control of release rate and contain physicochemical property of drug like pH solubility profile of the drug. For class 2 drugs, the systems that are developed for class 2 drugs are based on micronization, lyophilization, addition of surfactant, formulations as emulsions and microemulsion system, use of complexing agent like cyclodextrins. For class 3 drug, the class 3 drug requires a technology that addresses to fundamental limitation of absolute or regional permeability. Peptides and protein constitute the part of the class 3 drugs, and the technology for handling such material are on rise nowadays. Class 4 drug presents a major challenge for the development of drug delivery system, and the route of choice for administering such drug is parenteral, with the formulation containing solubility enhancers. Conclusion: this is all about the BCS classification of drug, its role, benefits, and application towards formulation development, which are being faced by the formulation scientists in the various pharmaceutical industries and the academic research. Thank you. Thank you for the patient listening.